hope I'm streaming now. Apparently, I think the Wi-Fi failed about 10 minutes into the class. So I will start the brachial plexus dissection video again. And once it's over, we'll switch to histology from my PPT. Okay, so then we won't depend on the Wi-Fi. Yeah? Yeah? Okay, seems to be working. Here we go. So I'm just starting the dissection video right from the beginning.
Brachial plexus. Let's learn about brachial plexus. Introduction. It is a plexus of nerves present in the axilla and forms the major nerve supply of upper limb. Formation. It is formed by ventral primary rami of lower 4 cervical spinal nerves and first thoracic nerve. Parts. It consists of roots, trunks, divisions and cords. Description of each parts. Roots. The roots of brachial plexus are 5 in number, from 5th cervical spinal nerve to the 1st thoracic spinal nerve. They emerge between scalenus anterior and medius muscle. The trunk of brachial plexus.
So just to orient you, this is the neck region. This is a strong muscle called sternocleidomastoid, and behind there's an edge of a muscle called trapezius, which is going to your scapula, and between these two and the whole bony clavicle there is a triangular area which we call the posterior triangle. Inside of that there is an upper triangular area which is mostly muscular in nature, and a lower triangular area, this one, small triangular area, which is very nervous in terms of the number of structures which are appearing there. So just orienting you to the anatomy, you're looking at the ventral surface, the anterior part of this person. This is the skin of the upper part of the chest. We've cut it to reflect the skin of the neck area. We've actually raised a flap from chin to clavicle. We open the skin like this, like a window, right? And this area is further divided by sternocleidomastoid. So we are ignoring the front part, we are ignoring the back part, and that middle triangle part is all that we are looking at.
This is the upper trunk of brachial plexus. Then this is the middle trunk, and then we have the lower trunk. These are the upper, middle and lower trunks of brachial plexus. Upper trunk of the brachial plexus is formed by ventral primary rami of 5th and 6th cervical spinal nerves. Then the middle trunk of brachial plexus is formed by the root of 7th cervical spinal nerve. Then the lower trunk is formed by the fusion of ventral primary rami of 8th cervical spinal nerve and 1st thoracic spinal nerve. In the subclavian triangle, this is the region of Erb's point. Erb's point is formed by meeting of the 5th and 6th cervical ventral primary rami, and then 2 divisions, anterior and posterior, and 2 important branches. Erb's point is a point in the upper trunk of brachial plexus where these 6 nerves meet or diverge. This is the suprascapular nerve which passes behind to the scapular region to supply infraspinatus and supraspinatus. The second branch from the Erb's point is nerve to subclavius. This will supply the subclavius muscle. Other nerves encountered in posterior triangle are the dorsal scapular nerve. This is the dorsal scapular nerve arising from roots of brachial plexus and supplies levator scapulae muscle. This pierces scalenus medius. We can see the scalenus medius here. This is the dorsal scapular nerve piercing the scalenus medius and this is the scalenus anterior muscle. The cords of brachial plexus are arranged.
Now you can see that we have extended the dissection area, right? Now here you can see the neck is closed, so there's a different cadaver, or maybe it's the same. No, the flap of the neck is closed. So here the flap is from clavicle, mid-sternal vertical incision, then across the costal margins, across the ribs, horizontal incision, a mid-arm horizontal incision, a vertical incision along the outer border of the deltoid, and you're connecting this skin flap here across the sides, so to give a full block. Now that's not how you will do it when you do the dissection. There will be only a axilla which you would have exposed, because it is a continuation of the pectoral region dissection which you would have done. So this was done like a specific window dissection for brachial plexus alone, right? You will be doing in continuity with the mammary gland region, the pectoral muscle region, and so on and so forth. So that's how the continuity will be inside your dissector manuals. Don't get confused. Just pay attention to this video from the point of all the illustrating it is doing for things which you have been memorizing only as a memory, verbal memory.
According to their name, around the second part of axillary artery. This is the second part of axillary artery. It is related to the three cords of brachial plexus. This is the lateral cord. This is the medial cord. And this is the posterior cord. Branches of brachial plexus. Branches of brachial plexus are divided into supraclavicular branches and infraclavicular branches. Infraclavicular branches. From the lateral cord, 3 branches. First branch is the lateral pectoral nerve. This nerve pierces clavipectoral fascia and supplies the pectoralis major and minor muscle. The second branch is musculocutaneous nerve. Musculocutaneous nerve pierces the coracobrachialis muscle. Musculocutaneous nerve supplies the coracobrachialis muscle before piercing it. The third branch of the lateral cord is the lateral root of median nerve. This unites with the medial root of median nerve to form the trunk of median nerve. Branches from the medial cord. Medial cord gives 5 branches. This is the medial root of median nerve. Then we have the ulnar nerve. Then the medial cutaneous nerve of the forearm. Then the medial cutaneous nerve of the arm. And then the medial pectoral nerve which supplies pectoralis minor. Posterior cord gives 5 branches. This is the main posterior cord. This is the upper subscapular nerve which supplies upper part of subscapularis muscle. This is the lower subscapular nerve which supplies lower part of subscapularis and teres major. This is the thoracodorsal nerve. Thoracodorsal nerve arises in between upper and lower subscapular nerves, passes along with the subscapular artery and supplies latissimus dorsi. Then we have the axillary nerve. Axillary nerve lies behind the third part of axillary artery and on the lateral side of radial nerve. The axillary nerve passes along with posterior circumflex humeral vessels through quadrangular space. This is the radial nerve. Radial nerve is the continuation of posterior cord of brachial plexus and the largest branch of brachial plexus.
Okay, so that was basically to orient you to how the dissection would have happened if you had been in the lab, but we will do it for you. There may not be time for you to do it, we will prosect it, keep it ready, and demonstrate it all to you when you come to campus. So please access these lessons if you can. There are a few free sample lessons, I think, on that website, but otherwise you can purchase the whole first year bit or something. It's available if you want. Otherwise, you can browse for similar things from other authors. Many anatomists have created their own dissections and also voiced the class for them. Please parallelly access these, because doing them separately from the theory won't work. Okay, so when the class is now, when lower limb is going on, whenever I have an opportunity, I'll try to share the lower limb dissections with you, but I don't have them downloaded on my system, and we won't get live net access all the time during the class time. So I have a challenge here. So that's why I'm just directing you to the website and hoping that at least some of you will be able to make use of the opportunity. Now what I'll do is open up my PowerPoint we have about 15 minutes. So I just wanted to show you some slides of the tissues that we had covered so far. I don't know how many of those classes are finished. For general histology, the last time we did lymphoid tissues and lymphoid organs, 4 types. And I have covered vascular tissue as part of circulatory system, but as tissues, as histology, you haven't done it. Then we talked about integumentary system, the skin, but we haven't seen it as a tissue set, which we see as 2 slides, thin skin and thick skin. And we talked about nervous tissue, neurons and glia, but what you see in the practicals are different slides, because we use different stains to show you the peripheral nerves, and then to show you the ganglia, and then to show you the basic neurons. Basic neurons we will see when we do CNS, cerebrum, cerebellum, spinal cord. So the way the practical goes is slightly different, but I'll try to show you some PPTs I've got. One sec. I think we can start with vascular tissue. I'll try to cover it as much as possible today. So we had said that the basic structure of all vessels, I think I had mentioned this when we were talking about the general anatomy classes, the circulatory system. So all vessels, whether it's the smallest capillary or the biggest arch of the aorta, has got 3 layers, and a vessel is a tubular structure with a lumen, a wall and a lumen. So the wall will have an innermost layer which is in touch with the blood which is flowing through the vessel, which is called the tunica intima, which will have an endothelial layer, a subendothelial layer, and an internal elastic lamina in the arteries. Then it will have a tunica media, which will be mostly muscle, arranged with concentric layers of smooth muscle cells. The number of elastic fibers, reticular fibers, in between intercellular matrix of proteoglycans and of smooth muscle cells itself. The proportion varies depending on the type of vessel, and that's how we classify it. The external elastic lamina is available in the larger arteries, we will see. And then all vessels have a tunica adventitia also, especially the thick ones. The thinnest capillaries will not have the external 2 layers, but they will always have the tunica intima. Tunica adventitia will be an extra layer of fiber added outside, which is longitudinally oriented. The tunica media is concentrically oriented in the tube. The shape of the muscles and the collagen fibers and everything is concentric to the lumen, whereas in the adventitia, the orientation of the fibers is longitudinal along the long axis of the vessel. So how will you tell an artery from a vein? Can you see? The artery is thick-walled, seems to have a tighter, rounder lumen, whereas the vein is thin-walled and is much more wider lumen. Okay, these are artery and vein of the same organ. Usually they travel together in a vascular bundle into and out of the organ. Surprisingly, the vein is a much bigger dimension vessel. The tunica media is thicker in the arteries will appear round and the veins will be collapsed shape. Endothelial layer has a pleated appearance. I don't know if you can make out that it's a little bit of a zigzag kind of appearance. We'll see in a higher power. So the artery will appear like a little wavy kind of a appearance of the endothelium. The insidemost layer is called the endothelium, which is simple squamous epithelium lining the lumen. Whereas endothelium of a vein will be smooth intercellular junctions with each other, no wavy thing. Then under this there is a subendothelial layer, the yellow. Then there is the tunica media, which is thick orangish muscle. This is only of course a painting. The smooth muscle, which is fusiform or spindle shaped cells concentrically arranged around the lumen. The arterial one will have much more muscle, thicker, and in between there these blue lines are basically a whole bunch of collagen and elastin fibers also contributing. Then you can see one more wavy line here, that's called the internal elastic membrane or lamina, which is a layer of elastin fibers, not smooth muscle. They're two different things. Smooth muscle is a live cell which contracts muscle, remember? Whereas an elastic fiber is an extruded protein created by a fibrocyte and thrown out there, and it has its own properties, and it just sits where it is and has a recoil function, so that if it's distorted, if it is stretched, it regains its original length. So that kind of tissue is laid down in this internal elastic lamina, which becomes visible or noticeable under the microscope, and that shows that this is an artery. In the vein, it may be there may be a little elastin laid down there, but it won't be, it's so scanty that is not at all significant, whereas in the artery it is. Similarly, now why are we calling it internal? There must be an external, others will just say elastic lamina, isn't it? So there is an external elastic membrane outside of the muscle layer also, but in this case it doesn't seem to be a continuous thing which you can easily identify, but that's where its location is, outside of the tunica media muscle. And then outermost layer is the tunica externa, which is fibrous, not muscular. Only tunica media is muscular in nature. So I think we covered everything which is in this particular slide. So the arteries will have an innermost tunica intima, which consists of an endothelium, a thin layer of glycoprotein, which is called the basal lamina, a little bit of subendothelial connective tissue, and an internal elastic lamina. So these four things together is equal to tunica intima. The tunica media consists predominantly of smooth muscle and or elastic tissue. Outside, it is limited again by an external elastic lamina. Outermost adventitia is only connective tissue in which collagen fibers are very prominent, and remember the orientation is longitudinal, not concentric, and this prevents undue stretching or distension of that. When the blood is pumping inside or flowing inside under high pressure from the pumping action of the heart, the tendency is for the blood to push, and for the muscle and the elastin and everything to stretch. So this adventitial longitudinal fibers are preventing undue stretching or distension of the artery. So endothelium, basal lamina, subendothelial connective tissue, internal elastic lamina is equal to tunica intima. Then tunica media is mostly smooth muscle and some amount of fibers, connective tissue fibers, including collagen, elastin, reticulin, but largely when they are there, a good amount of elastin will be there. Then there is also an external elastic lamina, which is part of tunica medium. Tunica adventitia is mostly connective tissue, longitudinally oriented, not concentrically oriented. Now, depending on the proportion of which tissue is predominating, we divide arteries into two types, elastic and muscular. So if tunica media has lot of elastin, it's an elastic artery. If tunica media has lot of smooth muscle, it is muscular artery. Okay, the ones which are closest to the heart and have to distend the most to absorb the pressure, to be able to take the pressure of the heartbeat, are elastic arteries. All right, they are the largest vessels. They supply the head and neck and the proximal limbs, subclavian, common iliac arteries. All the remaining arteries are muscular. Branches of elastic arteries are all muscular arteries. Carotids, brachials, femorals, mesenterics, all of them are muscular arteries. So an elastic artery will show a smoothish lumen. It doesn't have that wavy look so much. It doesn't show any lamina at all because there is so much elastin in the media that you can't differentiate it from an internal lamina, external lamina. The elastin of the inner lamina, outer lamina, and within the media itself are all looking continuous with each other. They're not very well distincted from each other. And the adventitia is quite a bit, doesn't take too much of a stain. So an elastic artery, if you put it under a very high pressure, you will see that there is a little bit of a waviness of the lumen. Internal elastic lamina not sharply defined. The media is broad, forming 2/3 of the thickness of the wall. When you stain it, not with hematoxylin eosin but with elastin, you see some fibers which the muscle fibers, the muscle cells, no other tissue stains except elastin, and that stains dark. So you're seeing how much of elastin is there in an elastic artery. So this is a good book if you get your hands on it. Vitas Functional Histology, it's a good textbook. So internal elastic lamina, media, and so much of adventitia. External elastic lamina is already here, and all of this is adventitia. Whereas now this has been stained with a different stain. Homework, find out what could this stain be where your elastin is showing up as reddish. Which kind of stain will make it reddish? Okay. Whereas in a muscular artery, because the media is full of muscular tissue, the inner and outer elastic lamina will be better defined and thrown into folds. And you see that the lumen is looking very wrinkly because of those folds. The internal elastic lamina is very prominent. This is RBCs inside the lumen. Okay? And adventitia is less. Thickness of adventitia is less. Thickness of media is lots. The endothelium is not very clear, but the internal elastic lamina is throwing that endothelium into a baby form. So when you compare to the radius of the lumen, this is the center, from here to here is radius. The media is almost equal of that, and adventitia is much less. Right? So that would be roughly the muscular artery. This is a longitudinal cut to the same. This is a transverse section to the tube. If you cut it like this and open it out, you see the cut section. You see that the muscle cells have been cut in a different orientation, right? Here you're seeing dash-shaped nucleus because the muscle cells have been cut longitudinal. Here the muscle cells have been cut vertically because of the different way in which we cut the artery. Adventitia is very little, right? Media is a lot, and E is the endothelium of the lining vessel. This is much higher magnification. So if you stain it for elastin, you see this beautiful internal and external elastic lamina nicely outlined, and doesn't seem to be any elastin in between the smooth muscle fibers of the tunica media, whereas in the elastic artery there was a lot of elastin. We saw when it's stained when it's all black, right? Here in the media there is no black, but the two elastic laminae are very prominent. But we don't have these slides for you with elastin staining, special staining. We only have H&E slides, and that's how you'll have to remember them or find them. Okay, I have 5 minutes. I'll quickly go through the rest of the slides. A small muscular artery will be of a small dimension of 0.5 to 2 mm diameter, but the distinct internal elastic lamina will still be there, and you'll have 3 to 10 concentric layers of smooth muscle. Okay, so this is a smaller size where the whole artery you can see in one cut section. You're seeing an E for endothelium, you're seeing an M for media. So 2, 3, 4 layers of muscle, and hardly any adventitia. There doesn't seem to be any adventitia outside at all. It's almost all muscle, and little bit of adventitia is there on the outside. So the difference is the tunica media. In elastic arteries, the media is mainly elastic tissue, as many as 50 layers of elastic membranes. And muscular arteries, smooth muscles, arranged circularly, that could also be 30 to 40 layers in the bigger muscular arteries. Alright. Internal elastic lamina is not distinct in elastic arteries because there is so much elastin in the media itself. An arteriole is a branch of an artery, and the small muscular arteries give branches called arterioles. The transition is gradual, but the reduction in number of muscle layers is the main point, and arterioles are defined as all arterial vessels which are less than 0.5 mm diameter, and they will not have an external elastic lamina. Okay, there will be an internal elastic lamina, but it will be thin and fenestrated. In the most terminal arterioles, even that one will be absent. So if you look at arteriole, it's beautiful and lovely little picture. So in this, what you're seeing is the endothelium is the innermost nuclei of the endothelium. TM is tunica media. I don't know what A is arterial lumen, and V is venous lumen. It seems to have something in it. Some blood has stayed with RBCs. It's a lovely magnification. This is the endothelium of the venule. This is the tunica media of the venule. This is the tunica adventitia of the venule. So it's looking almost as if it's merging into the surrounding tissues, right? So what you will basically look for is the endothelium and an empty space. A lumen will be pretty clear in an arteriole because of the roundness, because of the muscular tone. Whereas in the vein, there is not much muscular tone. Firstly, there is not much muscle in the wall, so it tends to have a collapsed or a shapeless kind of a lumen. In this one, it seems to be more rounded, but this one is the artery and this one is the venule. The lumen is much bigger. There is no wall. Here, the lumen is smaller. The wall is thick. Okay. Capillaries are the smallest, and they are only having tunica intima. They don't have anything else. No media, no adventitia. And the diameter is similar to the RBCs. I mentioned this before. And outside of this single layer of flattened endothelial cells, there are extra cells called pericytes, which embrace the capillary endothelial cells. So this is a capillary, right? So, and RBCs are going through it one at a time. There's no space for more than one RBC at a time. That's a lumen, all right? And RBCs are inside. And this is the external tissue, whatever tissue that we have taken it out of. Okay. Continuous capillaries are found in muscle, nerve and connective tissue, whereas fenestrated capillaries, you remember when we talked about endocrine organs? We said this is an important place where you get to see fenestrated capillary. That means there are holes in the walls, like instead of being a continuous plastic tube, it's like a perforated tube which allows large molecules to get in and out. Therefore, we need it where large molecules are either being secreted like pancreas or they're being absorbed like intestine. And endocrine usually is producing steroid molecules, big ones. So you need fenestrated capillaries to be able to accommodate the larger molecules which are negotiating the capillary from extravascular to intravascular. Sinusoidal capillaries we mentioned, especially in our spleen, lymphoid organ. We said they're discontinuous, the endothelial cells and basal lamina. The cell to cell junctions, they leave gaps. There are gaps. The cell is there and then there'll be a gap, naturally occurring gap between the endothelial cells, and this is available in bone marrow, liver, spleen, lymphoid organs and some endocrine glands, which allows good amount of molecular sharing between inside and outside. Plus the lumen is wider. We said the sinusoids are larger lumen than capillaries. So endothelial cells continuously with each other, flattened out cells with the nucleus, and this is a continuous capillary where this intercellular junction, the boundary is tight, right? And this is the basement membrane, and this is the wall, the cell membrane, right? Which is allowing small vesicles from in to out and from out to in, right? So basement membrane has to be penetrated for the molecules to be able to go in and out. Whereas in a fenestrated capillary, when you look at the intercellular space, basement membrane, etc., you see that there are perforations, there are holes where in between the cell junctions there are gaps. And so this boundary between endothelial cells, this is looking the same as this, but there are gaps, holes within the flat cell itself, right? And that's why they're called fenestrated capillaries, and they allow much easier exchange of secretions. This is how it looks like. Very beautiful electron micrograph from the kidney. Fenestrated capillary available in the kidney cortex. So can you see in the cell itself, between the outside of the cell and the inside of the cell, there is one passage in several places. So it's like having a double sandwich. Oh no, can't even call it a sandwich. Let's say slice of bread. That's also not a very good. Okay, 50-50 crackjack biscuit. Right? You have the cell membrane, you have the cytoplasm in between, you have a nucleus somewhere, and then through and through the cell you have holes. Okay, so those are fenestrae which are allowing things to go from one side of a flat cell to other side of a flat cell. This is the extravascular, this is the intravascular surface of that lining epithelium of the capillary. So it's just two cells. There's one cell, and then there's a junction, and then this whole thing is one cell. Right? Two cells are forming the entire capillary periphery, perimeter. Right? This is a very high power. 2 microns. This much is 2 microns. So this much is 1 micron. Really high magnification to show you what a capillary cut section looks like. And you will see it like this in a stained specimen with HNE. And you will see pericytes. I don't know if they've shown you in this pericyte. They'll be here you are. So you have another cell, bigger one, with a foot process. Can you see? Which is holding this capillary. Right? So this pericyte is so big that it is almost holding half of the diameter of the capillary. So from here to here, it's wrapping the capillary. So a pericyte is surrounding the wall of a capillary and doing certain functions. We'll come to that in the specific organs in which we will show you. But its nucleus will be seen as an extra nucleus over and above the endothelial lining of the capillary itself. Venules, they are the smallest veins into which the capillaries drain, 20 to 30 microns. They also have an endothelium, a basal lamina, and a very thin adventitia, almost no media. And they will also have pericytes. A little bit of muscle may be there in some, and then we call them muscular venules. If there is muscle, it will offer resistance to the flow of blood, and veins are very low pressure systems. So the muscle in general is avoided in the structure of a vein. The connective tissue components are much more pronounced, little or no smooth muscle. So this is the vein, and they cut it longitudinally, right? So this was the inside of a vein. This is the long wall of the vein. It was like this, and you cut it like this and opened it out, and you can see on the inside surface, on the tunica intima side, there are soft tissue intima folds. You see, it's a fold, flaps, which are seen. I wish you all had done dissection, but then we would have opened the big veins and shown it to you. This is what the valves look like. When you see that valve in cut section, this is what it looks like. There is an endothelium tunica which is forming a flap, which just hangs there in the middle of the lumen, and this one is also a flap, just a single layer of cell flap, and it is allowing blood flow only in one direction. When this blood tries to go back, it will fall into this cup-shaped depression and make these two flaps touch each other. Right? So when blood is returning to the heart from the lower limb, it's flowing in this direction. So when it's coming up, it pushes these two flaps aside and flows up. But if it's trying to flow back, it fills up the cup, and the two sides, the edges of the cup approximate to each other, not allowing a reverse or a retrograde flow. Very important. So this whole thing, the whole cut section of a venule or a vein with a valve passing through some other tissue which this one's blood supply is going through. So a medium-sized vein will have a little bit of media and a lot of adventitia, and in the media it will have smooth muscle as usual. A large muscular vein will have an intima, a little bit of media, and a lot of adventitia. So the muscle is in the adventitia. This is a strange thing. Till now we said muscle is always in the tunica media. So the point to remember is the good bunch of muscle, smooth muscle, is there in the adventitia layer only in the large veins. That's why they're called large muscular veins. And the muscle may be oriented in a concentric fashion, that means around the lumen, and also in a longitudinal fashion. Along the long length of the vein, some amount of muscle may have been provided. Even then, it is much, much less than an artery. The amount of muscle and the muscular action is much less than in an artery. So, artery had internal elastic lamina. First it had an endothelium, then it had internal elastic lamina, then it had tunica media, then it had adventitia. See the proportion. Vein has the same endothelium, same media, same adventitia, but look at the proportion. Small vessels which serve larger vessels are branches which come and nourish the outer median wall. When you have a wall which is so thick, the blood inside is obviously oxygenated and nourished. So via diffusion, a good amount of the thickness of the wall of this vessel can be kept alive. But from here onwards, it's not sufficient to be managed by diffusion. So you need branches of branches of branches of branches, fine branches which come back and penetrate the adventitia and keep it alive. They perfuse the adventitia and outer media to keep it alive. So the aorta will have its own vessels. The subclavian will have its own vessel. You come to lower limb. The femoral will have its own vessels. They are called vasa vasorum. Okay, so this is the media of a large artery. When you go into, okay, it's already adventitia. When you come into the looser area and you go towards the outside, you're seeing some small vessels embedded in between the connective tissue layers of the tunica adventitia. These are the vasa vasorum. So the differences between elastic and muscular arteries, large veins and medium-sized veins, venules and arterioles, penetrated capillaries and continuous capillaries. I'm sure this is there in one of your books. Please find it in your histology book so you can make out. In the practicals, we will be showing you 4 slides: elastic and muscular artery, large vein and medium-sized vein. And you have to be able to differentiate. Firstly, identify this is vascular tissue. It will be a round section of a tubular structure, right? And secondly, identify which of the 4 it is. That is what you have to do in your practice. Okay, I'll close now because we are 11 minutes out, and I will continue with some more histology in the next class. I hope to show you some more dissection, but if I cannot, you please find that resource and access it. Okay, thank you.